Right now, there are mainly two ways to deal with viruses: vaccines and antiviral medication. Both of these methods involves an injection or ingestion of some kind to either help the body's immune system or to disrupt viral processes. But various research shows how they can be inhibited with visible light using a method called impulsive stimulated Raman scattering, or ISRS. To understand how this fun method of virus destruction works, we must first know a little bit about resonance frequencies and viral anatomy. When disturbed, matter will vibrate at specific frequencies. The frequency at which a particular object likes to vibrate at is known as its natural frequency. This natural frequency is determined by factors such as the shape and material composition of an object. When an object is disturbed, it will emit mechanical waves, like sound, at its natural frequency. In fact, this is how many musical instruments function. For example, the sound produced by a bell when it is struck is its natural frequency. Resonance occurs when an object experiences a forced vibration from an external source with the same frequency as its natural frequency. The constructive interference causes the amplitude of the object's vibrating to greatly increase, making it vibrate much more violently and with much more energy. A great example of this effect in action is the ability to break a wine glass using sound. If a wine glass experiences enough stimulation by an external sound at its resonance frequency, the vibrational energy of the glass will become too high and it'll cause the entire wine glass to shatter. In this great video from MIT taken with a high-speed camera, you can see just how violently the glass is vibrating before it shatters. Keep this all in mind for later. Now that you know about resonance, let's do some basic viral biology. Viruses are essentially a small bit of genetic material, either RNA or DNA, enclosed within a shell made of protein, or a capsid. On their own, they are completely useless little things, and can only function if their genetic material is able to enter a living cell. Once inside a cell, they hijack the cell's protein construction mechanisms, turning it into a factory to produce more viruses from the virus's genetic material. After enough viruses are made, the cell is no longer able to contain them all, and it explodes, releasing a small army of viruses to go off and infect more cells. Here is where this application comes into play. By analyzing a virus's protein capsid, its natural frequencies can be determined. From there, the capsid can be bombarded with pulses of light, calibrated to the frequency of the protein. Because light travels in waves, the resonance effect of the light causes the protein to vibrate much more violently, and with enough vibrational intensity from the light, it can be damaged or destroyed. Remember the footage of the wine glass? That's essentially what's happening here. With a damaged or destroyed capsid, a virus can't enter a cell properly, and if exposed, its genetic material, such as its RNA or DNA, can quickly break down and become useless, destroying the whole thing. Most of this research has been done on a bacteria-killing virus called the M13 bacteriophage. Research by Kongtong Sen shows that the low-frequency vibrational modes, or the lower natural frequencies of the M13 bacteriophages, are associated with greater capsid damage from the axial torsion due to the vibrations. In his experiments, M13 bacteriophages could be inhibited with a light of a wavelength of 425 nanometers being pulsed at 100 femtoseconds, and for reference, a femtosecond is 10 to the minus 15 seconds, so these are incredibly fast pulses, and at an intensity of 49 megawatts per centimeter squared. Well, this seems like a very fun way to prevent and cure diseases. There are still several drawbacks to this method. One challenge is finding the natural resonance frequency of a virus capsid. Because virus capsids are composed from many different molecules and atoms, each with their own natural frequencies, it can be difficult to determine what's the most prominent low frequency. Furthermore, this is yet to be performed on viruses inside another living organism, and the technique would likely need some modifications to accommodate for the new challenges of barriers such as skin and living tissue getting in the way of the light. The fact that visible light can be used to stimulate capsid proteins also opens up the possibility for other mechanical waves to be used, such as sound. Unlike antiviral medication, which can sometimes cause side effects for certain people, the frequencies used to inhibit these viruses are far too weak to be harmful to any human cell, and this opens up this technology as a potentially powerful way of saving lives. While this method is not yet ready for any kind of clinical use, it remains a super cool way of dealing with viruses, and I look forward to its continued development. If you would like to learn more, then please consider reading the sources in the description of this video. They go into everything in far more detail than I could ever hope to. Thank you for watching.